So good morning, everyone. My name is Yoyo, and I'm a student in Honors Independent Research this year. And my mentor is Dr. Chen. And today I'll be talking about developing an antibiotic propolis cream against methicillin-resistant bacteria in the LOI. So first of all, I live on campus as a dorm student and where the LOI is right next door. So whenever I go to Waikiki, I have to cross the bridge over the LOI and then watch people paddling in this canal. So then I just start to wonder and then research more about this canal. And then I realized that this urban marine estuaries, such as the LOI Canal in Honolulu, they're often impacted by microbiological contamination that affects human health. And these contaminations includes bacteria and bacteria contamination like MRSA, which stands for methicillin-resistant Staphylococcus aureus. And as you can know from the name, that this is methicillin-resistant, where it's antibiotic resistant, and therefore it is very important for us to look closely into it because using a lot of antibiotics could cause a lot of problems. But firstly, let's talk about antibiotic resistance and how does it occur. Well, first of all, there's a bunch of bacteria and only a few of them are resistant to antibiotics. And then that's when the antibiotics come in and kills the germs, and no matter if they're pathogens or not. And after that, the remaining are the resistant pathogens so that they can develop freely. And finally, the bacteria transfers resistance to other bacteria by reproducing. Now, why is this so important? Well, because as you can see from this process, the more antibiotic you use, the more resistance there's going to be built on your body. And therefore, if you want to prevent infection from bacteria like MRSA, you don't want to use antibiotic all the times. And that's when I'm introducing this natural substance that is propolis to help people to prevent infection, as they also has antibiotic properties. So you might be curious what propolis is actually, propolis actually is. Well, it's a mixture of substances used by the bees to defend the hive. Whenever there's an intruder into the beehive, the bees would use propolis to mummify the bodies in order to prevent infection from the dead body to other parts of the beehive. And it is bee glue, it is sticky and resinous material released from various plant sources and is responsible for various bio activity, such as antibacterial activity. As you can see from the pie chart, that 50% of the composition of this propolis are plant rosin, and only 5% are the organic substances. And actually, these 5% of the organic substances are responsible for its antibacterial property. And the picture in the middle, the brownish substance in the spoon are the propolis, and this is how it looks like. According to researchers, propolis is more effective against gram-positive bacteria compared to gram-negative bacteria. And one way to determine if this bacteria is gram-positive or negative is by staining it. And after the staining, those gram-positive bacteria will show purple as you observe it under a microscope, and those bacteria that are negative will show up as pink. And as I have mentioned before, a, a big propolis is formed by collecting substances from different plant sources. And so therefore, its property is it's very much dependent on the location where it was collected. And so here to the left of the screen, you can see that these are the antibacterial activity of propolis from propolis collected from different places. And you can see Argentina propolis and also the Chinese propolis are the most effective. However, little research have been done on the propolis here in Hawaii, and therefore I think it is really interesting to look closer at the Hawaiian brown propolis and see how this local propolis could help protecting the health of the paddlers or people who interact with the water body that has been contaminated. And in my research, I used hardy chrome MRSA plate, which is a selective and differential culture medium that facilitates the isolation and identification of methicillin-resistant Staphylococcus aureus. And it has a chromogenetic substrate in the plate that could change the color of the bacteria. So after you plate the bacteria onto the plate, those bacteria that are MRSA would turn purple, and those other strands will turn, or other methicillin-resistant bacteria will turn blue on this plate. As you can see on the plate in the middle, well, maybe the color is kind of hard to see from here, but it'll be really easy to distinguish between the blue and purple color. So the first part of my project, my initial goal was to test the effectiveness of Hawaiian propolis on MRSA, and that's why I was using the hardy chrome MRSA plate. So I first had to collect sample of water from alloy, and I concentrated the bacteria and plated them on this MRSA plate. And then I tried to isolate single colonies and extracted the DNA, did PCR, and then sent the PCR samples to do sequencing. However, the sequencing result didn't come out to be what I was actually looking for, which is MRSA. The the results that came out was actually a kind of fish disease. But I was pretty sure that the colonies that I did the PCR was the purple colonies, which means that this route is not a good way to continue. It was a really bad news to me, but then we quickly decided to go the other way, which is to develop a cream that that could combat the methicillin-resistant bacteria instead of MRSA, because now we know that all the bacteria on this plate are methicillin resistant. Therefore, I came up with my new hypothesis, which is the propolis extract in cream can effectively reduce the growth of methicillin-resistant bacteria that adheres to surfaces in the water. And as a part of my hypothesis, which says the surfaces in the water. So the first step, I had to test out the different surfaces and to see which ones are the best to let the bacteria to adhere. So I tested chicken skin and filter paper and also erasers. So I put them into a lobster cage and put them in alloy for 2 hours. And after that, I took them out of the water and plated them on the plate and see how does the bacteria grows or how does it adheres to the surface. And as you can see on the picture there, of the tiny dots on the plates are the bacteria colonies. And the overall number of bacteria colonies on the chicken plate is like there are more bacteria on the chicken plate, and therefore we decided to use chicken skin or turkey skin to continue our research. So to test the propolis, we started with extracting the propolis. Well, we first collected a Hawaiian brown propolis from a household here on Hawaii, and we grinded them into fine powder and then mixed it with ethanol. And then the mixture was put into a shaker for 48 hours. And after 48 hours, we took out the mixture and we let it air dry in a hood for at least 3 weeks. And after that, the extracts were then reconstituted with DMSO, and then it was mixed with the cream. So here, the picture to the right is the picture of the mixed cream. And then now we have the cream. So the next step is to test the cream and the turkey skin in the alloy. So I had 9 pieces of turkey skin in total, which you can see on the right corner of this slide. The first 3 chicken skins, which is the first group, has the propolis cream on it. And the second group of 3 has the cream and everything else, including the DMSO, and everything else but the propolis extract. And the third one is cream only. And I put them into the alloy water for 2 hours. And after 2 hours, I took them out and then plated the chicken skin on the MRSA plates. And then I incubated them and and after 24 hours, I counted a number of bacteria colonies on the plates. And here are my results. So I counted both the purple colonies and the blue colonies because the purple colonies are indicated by the plates to be MRSA, but then I think it'll be better if I show like the different colors of bacteria on it. So the first graph shows the average number of purple colonies on the plates. The second graph shows the average number of blue colonies on the plate, and the third one shows the overall number of bacteria on it. So as you can see that the group that has propolis cream on it has the least average number of bacteria on it, which supports my claim that propolis in a cream could actually prohibit the growth of MRSA, oh sorry, methicillin resistant bacteria in the alloy and in a real setting. So why is this important? Well, it's because nowadays, as I have mentioned before, people don't want to use antibiotic anywhere in on their body. And this propolis cream would be a much safer and easier way for people to protect themselves. Whenever they are paddling or interacting with water, this would be a good way for them to protect, to like prevent infection of the bacteria. And therefore, I think it'll be really great to have this cream, and in the future it could be added. Propolis could be added into sunscreen or like other creams that people could apply easily when they decide to like paddle or do sports in the water. And now here are some limitations and some future researches. Well, due to time limit, I only did one trial, so I definitely need more replicates to 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 further confirm my results. And also I'm planning to compare different propolis um and see how does the effectiveness of these propolis differs from each other. For example, there are green propolis, um, red propolis, and also white propolis from Dr. Montgomery. And here's my takeaway from my research, and also my advices for future like other students who are uh, doing research next year. So one thing is that I suggest to always stay on top of your schedule and whenever you're planning something, make sure that you do enough research to make sure that you have the enough time to finish it, and also always make changes to your project and your method because not all the ways are going to work out. For example, I had to change my way because the first one didn't really work out. And there's one thing that that I would like to add on is to always be curious, which I just learned yesterday because you always have to like ask more questions and keep asking questions, and that's why you continue doing you keep like continue doing your researches. And after all of this, I would really like to thank Dr. Chen for your advices and your help. And I wouldn't like finish this without your help. And um I'm also like really thankful to like your patience and like figuring out everything with me. Yeah, so I'm really thankful for that. And I'm also really thankful for Mrs. Kobayashi for doing the bacteria staining with me yesterday, and also like going to the LOI and getting the samples with me. And thank you to Mr. Hill for taking care of my propolis extracts during the winter break. Thank you so much. And also to Sylvia because she came with me to collect my samples too. And last but not least, to Dr. Montgomery for providing me with the white propolis, which I'm going to do my further research and comparing different propolis in the future. And here are my references, and thank you for listening.